Hello, biologists. Welcome to the 2.4, 2.5 screencast. We're going to get going here talking about chemical reactions and enzymes. We'll start off real basic just by talking about reactants and products. So a chemical reaction is going to change a substance into something else by either breaking or forming chemical bonds. So sometimes when we have a reaction, we're going to break something down, and other times when we have a chemical reaction, we're going to put something together. Reactants are going to be the substances that are changed during the chemical reaction, and the products are going to be the substances made. So you're probably, or if you've seen a reaction, you are going to end up having your reactants over here on this side of the arrow. So reactants over here, and then they're going to form our products on the right side of the arrow. This arrow indicates a chemical reaction. So here's an example of a couple different reactions. I'm not quite sure why this box came out as a box. It's really supposed to be an arrow. That is the reaction. So here are going to be your reactants in the box, and over here are going to be your products. And then this is another chemical reaction. So there's an arrow showing the progress of the reaction, reactants over here, products over here. Alright, so many reactions in living things are going to be reversible. If it's reversible, you can see arrows that go in both directions. So the conversion, in this case of carbon dioxide and water into H2CO3 is going to be reversible, going in both directions. The concentration of products and reactants is going to stay relatively the same, and this is going to be known as chemical equilibrium. So equilibrium, you kind of reach a stable point, and the concentrations are going to remain the same. Alright, when we talk about chemical reactions, we have something called bond energy. That's the amount of energy required to break a bond. Sometimes energy is added to break bonds, or when energy is added, you're going to break bonds, and then when energy is released, we are going to form bonds. Okay, so the amount of energy required to break that bond is called bond energy. Before a chemical reaction can begin, you have to add energy to the system. This is called activation energy. You're activating it. You're starting it up. When enough energy is added to the reactants, bonds are going to break and the reaction is going to start. So you can think of activation energy as kind of going up a hill, what needs to be put in. Once you reach the point, the top of the hill, then your reaction is going to start and proceed. Here's a graph that shows the activation energy needed, kind of what I was saying, to get the reactants to a point where they can go. And then the next slide we'll see some different types of reactions. There's Roxy again. Can you say hi? Give me a bark. Is that the nemesises? Is it the nemesises? Is that them? Get them. Give me a bark. Is it them? Oh, she is being shy. Alright, so we have two types of reactions, like I mentioned. Exothermic is the first one. Exo means that the reaction is going to release more energy than they absorb. So the reactants are going to have a greater energy than the products. There she is. Oh, she wants her ball. Here you go, Rox. Go get it. So the reactants, here is our activation energy, how much needs to be put in. Then we have our products, and you can see our products end up below this original reactant line. Okay, so this is total energy released. Okay, so it releases more energy than they absorb. And you can see here that's going to be a little different than the endothermic. The endothermic are going to absorb more energy than they release because the reactants have lower energy than the products. So if we look here, the reactant line, here's our activation energy, and this line now ends up being above where our reactant started. So the reactants have lower energy than the products. So by lower, we mean they're lower over here than the products over here. That is endo. That takes us to section 2.5 on enzymes. Enzymes are going to be a specific type of catalyst. Catalyst, really, you can use catalyst in everyday language, like be a catalyst for change means speed something up or do something. Any catalyst is a substance that's going to speed up a chemical reaction. Enzymes are a type of catalyst in living things, so they are going to, and they can be used in other places as well, but we find them in living things. What they do is they speed up the chemical reaction by decreasing the activation energy that's needed to put in to get the reaction to go. And when that happens, it's going to make it go faster. So if we look at the graph, let's look at the graph. Here are our reactants. Here is the uncatalyzed reaction and how much activation energy we would need to put in if we did not have an enzyme present. Okay? This line, the dotted line, this is our catalyzed reaction. So this one, you can see this activation energy is much lower. We don't have to put in nearly as much energy to get it to go and make our products. All right? Here's a question for you too. Looking at this graph, a little review of endothermic and exothermic. Is this showing an endothermic or an exothermic reaction? So the answer on that last one, that was an endothermic reaction shown on that graph. So like I said, enzymes are catalysts found in living things. They're needed for almost all cellular processes. If we didn't have all the enzymes in our body doing all the things that we need to do, we probably wouldn't be here because everything would take a really long time. We're just much more efficient, and almost every single thing that we do is the result of an enzyme. A lot of times diseases, disorders, different things are because there is a lack of a certain enzyme. An enzyme is misshapen. Something is wrong with an enzyme. So we'll talk about more as the year goes on. Enzymes are a specific type of protein. Remember when we mentioned that the proteins are the most varied group? Well, enzymes are a huge chunk of that protein group. And they function best in a small range of conditions. That means each enzyme has an optimal temperature that it works at. It has an optimal pH range that it works at. The enzymes in our stomach are designed to work in a very acidic environment and probably wouldn't function in our blood or different places, and vice versa. If we took enzymes that didn't belong in our stomach, they would probably unravel and not be able to work because that pH wasn't in their particular small range of conditions that allows them to work. And as you can see in these pictures too, remember proteins. Proteins are that huge jumbled 3D mess. That's exactly, because it's a protein, this is what enzymes look like. So to simplify things, we draw an enzyme more like this over here. We have different spots on the enzyme that have different names. So the enzyme itself is this purple thing right here, okay? The substrate is what the enzyme is going to act on, okay? So in this case, or in all cases of enzymes, enzymes are going to have what are called active sites. Active sites are where the only place on this 3D structure, remember, it's that whole big jumble. It doesn't look like this nice, neat purple block. But the active site is the only spot where the substrates are going to be able to sit down, and where, remember, the whole goal of an enzyme is going to lower the activation energy so that the reaction goes faster. And part of what it does is it helps to facilitate the bond breaking and bond forming so you don't have to put in nearly as much energy. So enzymes function with this cool lock and key model. If we go step by step, this is kind of step number 1, this is step number 2, this is step number 3. Step number 1, the specific enzyme is going to bind to the active site. So remember, here is our active site, here are our substrates, this is the enzyme. The enzyme is going to join the substrates, weaken the bonds so that that activation energy is lowered, and we can make this product. In this case, we're putting something together. So we took these two separate pieces and we're putting them together into this bigger chunk. The product is going to be released, and one key thing about enzymes is they can be used over and over and over and over and over again. So they're very efficient. All right. All right, and then we are at our last slide, talking about, again, kind of going back to that point on the enzymes with their optimal temperatures, pHs. If there's some sort of disruption in homeostasis, it can interfere with the function of an enzyme. So any protein, we haven't talked about this yet, but this is a key word. You got to remember this one. Denature. Denature means to unravel, lose its shape. Remember, the protein, including enzymes, have this 3D structure where they are put together. And let's say you raise the temperature by 10, 15, 20 degrees, and it's not optimal for that enzyme. It can unravel, lose its shape, and turn into this, not nicely wound up 3D structure. Okay, so this is called denaturing. This is one of the things that we worry about if you were, as an adult, to get a really, really, really high fever. Temperatures of 104, even 102, 103, 104 are dangerous for an adult, because one thing that can happen is proteins start to denature, enzymes start to stop doing the things that they do for all of our body functions, organ failure, and it can be deadly if not treated. So that is one of the things that can happen. We'll talk a lot more about denaturing and protein structure as the year goes on. We'll learn how these proteins, here's a good little review of proteins. Remember, proteins, the monomer is that circle in your head. Think of what the monomer is. Amino acid, good job. And then we form this chain. Chain is the polypeptide chain. And then that's going to fold up into this 3D structure. And what happens if we have too high temperatures, not the right pH, it can go back into this. Actually, this is called the primary structure of an enzyme. Going back to what we talked about with like primary, secondary, tertiary, quaternary, when we talked about the levels of trophic levels or on a food chain. We use those same words to describe protein folding. All right, so there was a lot there. That does it for the notes for Chapter 2. We will run down all of this stuff on Monday and be ready for the test Wednesday. Great job, biologists. Roxy says, she's just laying on the floor right now. She doesn't really have a message.